Hi, today we are going to present our new shiny application, ConoServerParser, which is a complementary tool for filtering, exploring, and exporting Conotoxin sequences from ConoServer. So, what is ConoServer? ConoServer is a trusted, expert-curated database of conopeptide sequences and structures maintained by the Institute for Molecular Bioscience, Australia. It was created in 2007 and is led by scientists David Craik, Jan-Christoph Westermann, and Quentin Kaas, alongside their team of contributors. The name of the server Cono derives from cone snails, marine gastropods of the Conus genus, the subject of the server. Cone snails produce complex venoms composed of thousands of bioactive peptides. These compounds, known as conotoxins, are primarily used to capture prey, defend against predators, and deter competitors. The venom composition reflects their ecological diversity, targeting specific ion channels, receptors, and transporters in their prey's nervous or muscular systems. These conotoxins target neurological and muscular receptors with remarkable specificity, making them valuable for drug discovery, neuroscience, and evolutionary biology. Why is ConoServer so important? Given the rapid growth in conopeptides discovery and their complex classification, researchers rely heavily on ConoServer. What makes ConoServer invaluable is its rigorous, manually curated content. It integrates data from peer-reviewed publications, public databases like UniProt-KB, NCBI Nucleotide, and the Protein Data Bank, PDB. Curation includes analysis and annotation of sequences, classification into gene superfamilies, cysteine frameworks, and pharmacological family. Besides, ConoServer provides updated statistics, a list of documented post-translational modifications, a tool to predict and map signal and propeptides, allowing users to obtain the mature peptides, and it also has integrated a multi-criteria search tool, among other features you can explore on ConoServer. We have developed this shiny application, ConoServerParser, to enhance the user experience when exploring ConoServer information. This shiny application uses the ConoServer protein sequences as input. We are going to explore and filter in order to make it easier to obtain some sequences for other kinds of downstream analysis like machine learning, deep learning, weight, or to explore functionally related sequences from other venomous animals to these sequences provided by ConoServer. So now we are going to use this file, but you have the option to load it simply by clicking here. And we can see automatically that the application loads all the information. You can see here the number of entries, the total number of entries. You can see that we are parsing the information from this FASTA. You can see here we can show the header of the sequences, but now we are going to remove from here because we don't like to have these redundancies. So let's continue exploring this left panel. Okay, here we have this column selection for the table, which controls what information from the header will be parsed and shown here in this table. This is what the FASTA looks like. You have different pieces of information about the entry. ConoServer ID, the name, species, whether it's wild type or synthetic, the class, the gene superfamily, and here's the pharmacological family, the cysteine framework, and the source, whether it comes from protein level or nucleic acid level, and other related information. And here the sequence. What we want to do is parse all this information and represent it in the interactive table in order to filter and download exactly what we want to export, given the customized settings. Also, please note that sometimes some data may be missing, so we're going to classify it as unknown or unclassified, depending on how it's presented in the header. For example, if a field is labeled as unclassified, that's how we'll treat it in the table. You can also find in the metadata some labels or categories as unknown. So here we have the parsed sequence fields, ConoID, the protein name, the organism, which may be a species name or marked as synthetic. You'll also see protein type, conopeptide class, and more. you can select which of these elements you'd like to display in the table. Use this drop-down panel to select or deselect all. For instance, you can deselect all fields or select just one. As you make selections, you'll see that the number of entries in the table updates interactively down here. So let's select all again.
If you
You can also search. For instance, if you search for unknown, it will automatically filter the table to only show those entries. You can download the results at any time. I recommend always checking that all the rows you want are selected. To download the information in a tabular format, select the filtered rows. Use Show All to confirm everything's visible before downloading. Here you can export the filtered rows in different formats. Let's say you want to exclude certain organisms. It's easier to deselect them directly here, and then click on specific organisms or category. For instances, this option allows the user to exclude selected organisms. This is helpful when dealing with a long list and you only want to remove a few items. The filter section includes all categories or classification for each of the parameter displayed as column name, but these fields like protein type, including wild type, synthetic, precursor, and patented, are parsed from the FASTA header download from ConoServer. This is useful if you want to explore, for example, only precursor sequences. That classification is based on how ConoServer curators assign gene superfamilies. You can also apply this filtering steps to conopeptide classes or any other field, such as gene superfamily. The gene superfamily field supports various values, single-letter codes, full names, or divergent superfamilies. You can include or exclude unclassified entries here as well. All of this filtering is interactive. From a functional point of view, the pharmacological family is particularly interesting. We have 12 pharmacological families in total, and we can exclude unclassified ones if we want to focus on validated data. Every time you apply a filter, you can check here in the panel how many sequences match your criteria, per family, per superfamily, and with supporting evidence. This is particularly useful to check and guide you on what is going to be exported into FASTA files. You can also choose which separator to use for headers in the exported FASTA, comma, semicolon, underscore, or just preserve the original. You know this tab, where you can see the detailed statistics of the in-out FASTA downloaded from ConoServer. This preview here reflects the input file as parsed from ConoServer. The FASTA preview lets you check how many metadata elements are included in the header, in case you want to simplify it or only keep specific information like superfamily or pharmacological family. For example, you might select just ConoID, and you'll immediately see the header is updated accordingly. This is a head of the file or a preview on how the final FASTA file will look when downloaded, by clicking on these buttons or by exporting through these options. And this is an interactive way to have feedback on what is selected, filtered, and to be exported, and how the header is going to be composed. You can also generate plots based on the filtered data. You can reset all filters. This is the default reset button. Plots will update automatically. Let's say we plot superfamily versus pharmacological family. This allows us to explore how many sequences remain classified or unclassified. For instances, let's plot only the classified ones. Just deselect unclassified and the chart updates instantly. Now, imagine you want to study pharmacological families versus sequence evidence. Select what you need, and you'll see counts for each category. Let's plot only the 12 classified pharmacological families by removing here the unclassified. You can also check how many sequences are at the protein level, nucleic acid level, or predicted. And this functionality can be applied to the other fields, like protein type, if you want to study only wild-type mature peptides or cysteine framework. However, there are some things that can be improved, of course, but ConoServerParser could enhance the experience and reproducibility of ConoServer data. Indeed, there are some inconsistencies here. For instances, check this entry from ConoServer that we have in the downloaded FASTA, classified as omega Conotoxin, wild type. If I come here and load the FASTA containing all the information from ConoServer and search this entry, we can find it. But if we come to ConoServer and use this tool to find same entry, we can't find it. No results. Similarly, if we come here trying to search in the specific pharmacological family related to this entry, we can't find it. The thing is, I can find it here, but here this entry doesn't has any classification for pharmacological family, and this is an example of inconsistencies between the information shown in the ConoServer website among different tools and the information provided to be downloaded in the FASTA file. In ConoServer, we have this tool with this option to search a peptide and a change filters. You can apply some logic to customize the search. For instances, I check here to search all the pharmacological families, but well, it is showing everything instead of only the pharmacological families. So imagine I would like to show and export all the pharmacological families by selecting, not all, but all the listed families here to display the metadata, which can be helpful for downstream analysis. Well, I can select one by one, but let's select all. And I expect to have a button to download in .fasta, but we don't have it here. So let's select all here. Then we have this button to show all, but we don't have any option to download the selected sequences in FASTA format. So the option I found is to click on this button format. Then we need to copy and paste into text file and save it as FASTA, which usually can generate some mistakes. In ConoServerParser, we have deployed these options to download the gene superfamily and pharmacological family in FASTA by clicking on this button. The research years will have a .zip file containing 1.fasta per superfamily or pharmacological family. Decompress the zip file and check all the gene superfamily in separated FASTA file and with the pre-selected metadata, which is really useful to use as input in other bioinformatics tools for downstream analyses or biodiscovery. Well, now let's explore this interesting tool that predict the mature peptides of conotoxins and conotoxins-like precursors. Let's use this Y gene superfamily as example, since only have here 4 representatives, and if we select to show all, we find that we have 5 sequences, but it might be related with the lack of signal peptide in first one, which is a criteria for gene superfamily classification, but the tool can predict as well the signal of this sequences. Well, let's test with this one. Paste here. Notice that it generates an error, because in this section we can only use the sequence, without header. Then we have the prediction of signal, propeptide, mature peptides, which is interesting, and accurate information, but we still don't have the option to download in FASTA the resulting information. I mean the mature peptides. And if we would like to do a batch prediction, we need to use the other section by uploading the precursor sequences. As we mentioned previously, we don't have the option to download these sequences in FASTA format directly from a button, so we need to copy and paste in a text file and save it as .fasta. Then upload it. Run and wait. Then we will have the option to download the prediction in tabular format, having some description as column, such as entry, signal peptide, propeptide, mature peptide, among others. But still we don't have the option to download the mature peptides that we would like to use further in functional analyses for instances. So we also provide this tool that allow the user to upload the resulting .cvs file. Here is preloaded the Y gene superfamily, so you can test it easily here. The tool will parse the file, then replace the word precursor, if there with the word mature, and create the FASTA that can file downloaded, ready to use or re-uploaded, parsing in ConoServerParser, and adjust the elements to be included or excluded in the header. And that's all. You can find and test the tool at https://dannysario.shinio/ConoServerParser. You find it. Result. Thanks.